Now this is our 34 lecture, 34 number, and it is page 1. We are going to discuss mechanism of arrhythmia. Now, mechanism of arrhythmia, how arrhythmia is caused. In order to understand the mechanism of arrhythmia, it would be appropriate to know about the development of cardiac action potential. Now, regarding cardiac action potential, there is one point to remember, that generally action potential from any tissue is the same, always the same. The shape, the size, the timing, always the same. For example, action potential of nerve, it is like a spike, and it is whatever nerve you take, it will be the same shape, same time. Similarly, muscle action potential will be also similar, but heart has got a different types or slight different changes you will find in the action potential generated by the different part of the heart. There are basically two main types of cardiac action potential. One is slow. One is the slow response action potential. Slow response action potential. That is, it is observed in SA node and AV node. Then there is another type which is known as fast response action potential. It is seen in the structure like atrium, ventricular muscle, and Purkinje cells. So the two different groups, and among the group also there are variations. Now we will start from slow response action potential, that is arising from SA node and AV node. If you see, this is the shape of slow action potential. Slow response action potential or slow action potential. The resting membrane potential is minus 60 millivolt, and you will find that there are three phases, 0, 3 and 4. The 0 phase means depolarization. The 0 phase means depolarization. Phase 3 means repolarization. And phase 4 is pacemaker potential, pre-potential, or slow depolarizing potential. Three names. Now let us see here the speciality of this slow response action potential. Now, you know, if, for example, if a muscle, for example biceps, is supplied by musculocutaneous nerve, when you cut the nerve, the muscle is paralyzed. But if you denervate the nerve supply of the heart does not stop beating, it goes on beating without its nerve supply, independent of nerve supply. So now why it is so? It is so because of the phase 4. In other action potentials, you will find that the phase 4 is flat. Here the phase 4 is slowly depolarizing, and when it reaches to the level of minus 60 millivolt, then there is production of another action potential. This property is known as atomicity or rhythmicity. So why heart goes on beating without nerve supply is because SA node and AV node has got a phase 4 pre-potential or pacemaker potential. Now let us see what is the cause of phase 0. Phase 0 is caused by the entry into the nodal cells through L type of calcium channel. So zero phase is because of slow entry of calcium through L type of calcium channel. What is L type of calcium channel? This is long lasting voltage gated channel through which the calcium is going into the cell, causing slow causing not fast rapid depolarization, but a slow and a steady depolarization of phase 0. Then comes the phase 3. Phase 3 is due to efflux, that is potassium going out of the nodal cells through potassium channel. So potassium going out will cause repolarization or phase 3. Now let us come to the phase 4. Phase 4 is caused by influx into the nodal cell through T type of calcium channel. This T type of calcium channel is known as transient voltage gated channel. Now then another fact which causes the phase 4 is sodium influx into the cell through slow funny channel, sodium channel. So the pre-potential is caused by two things: by calcium entering into the cell, nodal cell, through the transient T channel, transient channel, and sodium entering into the node through a special type of sodium channel which is known as funny channel. So now let us analyze further. Two positive ions are going into the cell in phase 4, that is calcium and sodium. This slowly enters into the node, so there is a positivity inside, that is depolarization. When there is inside they becomes positive, it becomes depolarized. So slowly there is a slow depolarization in phase 4. So you see the slope. When slope reaches minus 60 millivolt, then a next action potential is generated. So this is why there is rhythmicity or atomicity. Atomicity depends on phase 4. Now another important thing which I told you, funny channel. Sodium is going through funny channel. Now you remember, sodium when it goes into the cell, it causes depolarization, and for depolarization then you get the action potential. Now here what happens, the sodium channel is entering while there is hyperpolarization. Sodium enters during the depolarization in all the other tissues, while here in phase 4 the sodium is entering slowly into the node when there is hyperpolarization. So this channel open during hyperpolarization, not in depolarization. That's why it is called funny channel. So this is what is the SA node. Now difference between SA node and AV node is not of shape. Shape will be the same. It is only the speed is slow in AV node. The duration will be the same, but the slope will will not be as is not steeper. It is flat in slightly flat in AV node. So difference between the SA node and AV node's action potential is they are similar and the causes are similar, except that the action potential generated in SA node is faster and it is slower in the AV node. Thank you, sir.